Good evening, students. So our today's class, the topic of our today's class is also motivated by question of a student, right? This student came after the class and asked from me, sir, how exactly pregnancy, some cases of pregnancy leads to diabetes, right? Now this is quite a genuine question, right? Quite a genuine and interesting question, right? Pregnancy is characterized by increase of progesterone, estrogen, and all the effects we know, right? The facial flushing, palmar flushing is because of vasodilatory effects of progesterone, right? Be it gastroesophageal reflux disease because that lower esophageal sphincter gets relaxed, right? All the effects which we see in pregnancy can be explained by these hormones, right? So how exactly diabetes, how exactly insulin resistance can happen in pregnancy, right? That's a very genuine and interesting question, and in this video we will try to understand how exactly some cases of pregnancy can lead to diabetes. The term we use is gestational diabetes mellitus, right? So let's try to understand it in a step-by-step manner, and it's an interesting mechanism. If you understand it, you will truly understand how various physiological mechanisms in our body interact and counterbalance each other, right? So let's start, right? So if we take the case of gestational diabetes, right, one hormone secreted by placenta is actually very important, right? And name of that hormone is human placental lactogen, HPL, right? This hormone is very important in pathophysiology of gestational diabetes, right? Now this hormone is, the molecular structure of this hormone is very similar to growth hormone and prolactin, both to growth hormone and prolactin. We can say homologous. Since the molecular structure of this hormone is very similar to growth hormone and prolactin, this means what? That this hormone can act on GH receptors and also on prolactin receptors, and this means it will have multitude of actions, right? It will also act on GH receptors, it will also act on prolactin receptors, so it will have multiple actions, right? And we'll try to understand these actions one by one, right? So this human placental lactogen is going to have facilitatory influence, right, on one very important hormone. That hormone is your hormone sensitive lipase, right? Hormone sensitive lipase. Now this hormone sensitive lipase will act on adipose tissue, right? It is going to act on adipose tissue. By acting on adipose tissue, it is going to increase the concentration of free fatty acids, right? So up till now it's simple. This HPL, human placental lactogen, increases in pregnancy. It is going to stimulate your hormone sensitive lipase. This hormone sensitive lipase will act on adipose tissue and it will increase the concentration of free fatty acid, right? Now what next? This human placental lactogen will also lead to serine phosphorylation of your insulin receptors, right? Insulin receptors right? They will undergo serine phosphorylation because of human placental lactogen. And since these insulin receptors are undergoing serine phosphorylation, it will start a cascade mechanism, right? It will reduce your PI3K recruitment, right? This recruitment will be reduced, right? Now, properly. Now this PI3K recruitment is reduced, so this is going to further reduce your AKT phosphorylation. Right? This will reduce your AKT phosphorylation. Now since this AKT phosphorylation is reduced, this AKT phosphorylation is actually very important for transport of GLUT4, transport channel which is involved in facilitated diffusion of glucose, right? So GLUT4 won't be transported, right? So GLUT4 translocation will reduce. Okay, if GLUT4 translocation is reduced, now what GLUT4 does, right? Suppose this is your cell, it can be skeletal muscle cell or smooth muscle cell. The GLUT4 actually transports glucose inside the cell, right? The glucose is transported inside the cell with the help of GLUT4, right? If GLUT4 is not translocated inside the cell, the glucose level in the blood is going to increase. Since the glucose is not going inside the cell now, the glucose level in the blood will increase. So you will have high glucose over here. Along with high free fatty acids, you will have high glucose. Right? Now what else? Right? This human placental lactogen, right, can also stimulate another enzyme, right? And that enzyme is your 11 beta HSD, right? 11 beta hydroxysteroid dehydrogenase, right? If this enzyme is stimulated, it will increase the concentration of cortisol. And as all of us know, if cortisol increases, your blood glucose will increase, right? And this glucose will also go in this pool, right? Now, apart from that, you saw this AKT phosphorylation is reduced, right? So this AKT phosphorylation is now going to upregulate your G6 phosphatase. Your G6 phosphatase will get upregulated. Right? If G6 phosphatase gets upregulated, the glycogenolysis will increase. If glycogenolysis increases, right, of course the glucose will increase, right? So this human placental lactogen can increase your glucose via various ways, right? Okay, and it can also increase your free fatty acids. It acts on hormone sensitive lipase, which acts on adipose tissue, and it increases your free fatty acid level, right? It acts on insulin receptor, the serine phosphorylation of insulin receptor, the PI3K recruitment reduces, AKT phosphorylation reduces, GLUT4 translocation is not happening, glucose can't come inside the cell, so glucose inside the blood increases and the overall glucose pool, it adds to the overall glucose pool, right? Now since the AKT phosphorylation, right, is reduced, it is going to upregulate your G6 phosphatase. So glycogenolysis will increase and it will also add to the glucose pool. Now, what's the issue here? If you get chronically high, right? If these two things stay chronically high, right, it will lead to a phenomena which we call glucolipotoxicity, right? This will lead to glucolipotoxicity. Right? Now this glucolipotoxicity can actually damage your beta cells, right? I will draw beta cell over here, and if you follow my previous videos, you know what is going inside these beta cells, right? So what is going inside this beta cell? The glucose is entering, right? And who is sensing the glucose inside beta cell? The glucokinase is sensing the blood, is sensing the glucose, glucokinase, right? It gets converted into glucose 6-phosphate, and after multiple steps, right, it will get converted into pyruvate, right? This pyruvate will go inside where? Mitochondria, right? And this mitochondria will produce what? ATP, right? The glycolysis will also produce ATP. Both these ATPs will combine and go where? They will go and bind to your potassium channels, right? When this ATP binds to potassium channel, the potassium channels inside these beta cells are ATP sensitive, right? So as soon as they close, the potassium will accumulate inside. If potassium accumulate inside, the potential inside will grow, will move towards zero, from minus 70 to minus 40 millivolt, right? At minus 40 millivolt, your calcium ion channels will open up, right? The calcium will come inside, and as soon as the calcium ion channel comes, these vesicles with the help of snare proteins are going to empty their contents outside. Like this calcium will go and bind to synaptotagmin, and the snare proteins will help in docking, right? So this much background we need to know, right? Then we will perfectly understand what glucolipotoxicity does to your beta cells, right? This glucolipotoxicity will reduce the level of glucokinase, right? It's going to inhibit the glucokinase, right? So this process won't proceed, right? This glucolipotoxicity will also lead to mitochondrial dysfunction. If mitochondria is dysfunctioning, of course, the ATP won't be produced. If ATP is not produced, it won't go and bind to potassium ion channels. If it doesn't bind to potassium ion channels, the potassium gates won't close, right? If potassium gates won't close, this change from minus 70 to minus 40 millivolt won't happen. Calcium gates won't open, right? And insulin won't be released. So this thing will also reduce the release of insulin, and glucolipotoxicity by causing mitochondrial dysfunction will also ultimately leads to reduction in the release of insulin, right? And glucolipotoxicity also damages your snare proteins, right? So it's like damaging all the steps, right? It is inhibiting your glucokinase, it is causing mitochondrial dysfunction, it is damaging your snare proteins, right? And it also leads to incretin resistance, right? So what are your incretins? That GLP and GIP, right? Through which mechanism they act? They act through adenyl cyclase mechanism, right? So this GLP and GIP, right? This glucolipotoxicity is going to inhibit this also, right? It will also lead to incretin resistance, right? So all the steps inside the beta cells, right, will be inhibited because of this glucolipotoxicity, be it glucokinase, be it mitochondria, be it snare proteins, or be it incretins, right? So what will happen if the beta cells, right, is undergoing so much hindrance at every step, right? This will lead to beta cell exhaustion. Right, beta cell exhaustion, and it will also lead to beta cell apoptosis. Right? Now, these are kind of pro-diabetogenic actions of HPL, right? Now, HPL has got two kinds of actions, and these two actions kind of balance out each other, right? Now, HPL, when it acts directly on beta cell, right, it actually causes proliferation of beta cells, right? When HPL, when it acts directly on beta cells, right, it acts via prolactin receptor, right? In short form, I will write PRLR. In starting, we discussed now that this human placental lactogen is homologous to prolactin, right? So it acts via prolactin receptor, right? And it activates the mTOR pathway. And by activating the mTOR pathway, it increases the beta cell mass by 2.5 to 3.5 times. Now here lies the catch, right? Just in case if there is any genetic anomaly, the body always keeps some beta cell reserve for emergencies, right? But if just in case there is not enough beta cell reserve because of some kind of genetic defect, right? Many research papers have found out that being from South Asian descent, right? If you are having South Asian genes, right, that itself is a risk factor, right? That you are not having enough beta cell reserve, right? Or if you are already having PCOS, right? So beta cells are already tied up, so they are not having enough reserve, right? So this thing, if there is not enough genetic backup, right? If your genes are defective and due to your genes you are not having enough beta cell reserve, right? Then this will lead to beta cell exhaustion and ultimately gestational diabetes mellitus, right? So just in case your genetic reserve is not there, right, this will lead to beta cell exhaustion and diabetes, and ultimately what we say, GDM, gestational diabetes mellitus, right? And what are the conditions which can predispose to it? Conditions like PCOS. You won't have enough beta cell reserve, right? And South Asian descent. If you are from South Asia, right? Then this will lead to beta cell exhaustion and gestational diabetes mellitus, right? So let's try to sum it up, right? So which hormone is mainly responsible for your gestational diabetes mellitus, right? The hormone mainly responsible is your human placental lactogen, right? What human placental lactogen will do? It will stimulate your hormone sensitive lipase, right? This hormone sensitive lipase will act on adipose tissue and it will increase the pool of free fatty acids, right? This human placental lactogen, right, will also act on insulin receptors and will cause serine phosphorylation of insulin receptors. This will lead to a cascade of reactions. The PI3K recruitment will reduce, the AKT phosphorylation will reduce. If AKT phosphorylation reduces, your GLUT4 translocation will reduce. Since the GLUT4 is not getting recruited now, the glucose won't be able to enter the skeletal muscle cells, right? If glucose is not entering the skeletal muscle cells, the glucose in the blood is going to increase. This will add to this pool. Your human placental lactogen can directly stimulate your 11-beta hydroxysteroid dehydrogenase. This will increase the level of cortisol, and this will also leads to increase in glucose level, which will add to this pool. The AKT phosphorylation over here, the reduced AKT phosphorylation over here will upregulate your glucose 6-phosphatase. This will increase the glycogenolysis. This will also add to the glucose pool. If both free fatty acids and glucose stay increased for a long duration, you will get a condition called glucolipotoxicity. This glucolipotoxicity will impair all the steps inside your beta cells. It will inhibit your glucokinase. It will cause mitochondrial dysfunction. It will also cause the damage to snare proteins, right? And it will also lead to incretin resistance. All these steps will reduce your insulin release, right? This much hindrance will ultimately lead to beta cell exhaustion and beta cell apoptosis. But HPL, when it acts directly on the beta cells, right, it acts via the prolactin receptors. It stimulates the mTOR pathway, right, and it leads to proliferation of the beta cells almost 2.5 to 3.5 times. But just in case there is a genetic defect and not enough beta cell reserve, right? The being from South Asia is also kind of risk factor. Having PCOS is also some kind of risk factor, right? So if there is any genetic defect and not enough beta cell reserve, then it will lead to beta cell exhaustion and ultimately gestational diabetes mellitus, right? So hopefully this video has helped you in understanding the various mechanism behind the gestational diabetes mellitus, right? I enjoyed reading about it, and hopefully you also enjoyed listening it. Okay, wish you all the very best. Thank you.